Welcome to the exciting world of gene editing. We are on the cusp of a new era in genetics, one where we can precisely alter the very building blocks of life itself. Imagine a world where we can cure genetic diseases, engineer drought-resistant crops, and even design new organisms with unique traits. This is the promise of CRISPR, a revolutionary technology that is transforming our understanding of genetics and opening up unprecedented possibilities for the future. CRISPR is short for Clustered Regularly Interspaced Short Palindromic Repeats. It may sound complicated, but it's actually quite simple. CRISPR is a tool that allows scientists to edit genes with incredible precision, like a surgeon operating on DNA. This technology has the potential to revolutionize medicine, agriculture, and even the way we understand life itself. In this essay, we will embark on a journey to demystify CRISPR and explore its profound implications for our world. We will delve into the science behind CRISPR, examine its real-world applications, and contemplate the ethical considerations that accompany this groundbreaking technology. Join me as we unravel the mysteries of CRISPR and unlock its transformative potential. Before we dive into CRISPR, let's first understand the basics of genetics. Our bodies are made up of trillions of cells, and each cell contains a complete set of instructions that determine our traits, from our eye color to our susceptibility to certain diseases. These instructions are encoded in our DNA, a long double-stranded molecule that resembles a twisted ladder. Each rung of the DNA ladder is made up of two chemical bases, called nucleotides. There are four types of nucleotides: adenine A, thymine, T, guanine, G, and cytosine, C. The sequence of these nucleotides determines the genetic code, which tells our cells how to build and maintain our bodies. Genes are specific segments of DNA that contain the instructions for making proteins, the workhorses of our cells. Proteins carry out a wide range of functions, from transporting oxygen in our blood to regulating our metabolism. Changes in our DNA sequence, called mutations, can alter the instructions encoded in our genes, leading to changes in protein function. These changes can have a wide range of effects, from harmless variations to serious diseases. CRISPR is a revolutionary gene editing tool that allows scientists to make precise changes to DNA sequences. It acts like a pair of molecular scissors, cutting DNA at specific locations, allowing for the removal or insertion of genetic material. CRISPR was originally discovered in bacteria, where it serves as an immune system to protect against viral infections. When a virus invades a bacterium, the CRISPR system captures a piece of the viral DNA and integrates it into the bacterium's own genome. This creates a genetic memory of the virus, allowing the bacterium to recognize and defend against future infections. The CRISPR system uses this stored viral DNA as a guide to target and destroy invading viruses. Scientists have harnessed this bacterial defense mechanism to create a powerful gene editing tool. By designing guide RNAs that match specific DNA sequences, researchers can direct the CRISPR system to cut DNA at precise locations. This allows for the removal of faulty genes, the insertion of new genes, or the correction of genetic mutations. The most widely used CRISPR system is CRISPR-Cas9. Cas9 is an enzyme, a protein that acts as molecular scissors, cutting DNA at specific locations. The guide RNA acts as a GPS, directing Cas9 to the desired target sequence in the genome. Once Cas9 makes a cut in the DNA, the cell's natural repair mechanisms kick in, attempting to fix the break. Scientists can exploit these repair mechanisms to introduce specific changes to the DNA sequence. For example, researchers can supply a template DNA sequence along with the CRISPR-Cas9 system. This template DNA serves as a blueprint for the cell's repair machinery, guiding the insertion of new genetic material into the genome. CRISPR-Cas9 has revolutionized gene editing because it is relatively simple, inexpensive, and highly efficient. Unlike previous gene editing technologies, which were often cumbersome and prone to errors, CRISPR-Cas9 offers a precise and versatile tool for manipulating DNA sequences. Section 5. The power of precision-targeted gene editing. The precision of CRISPR-Cas9 opens up a world of possibilities for gene editing. Scientists can target specific genes, even individual nucleotides within a gene, with remarkable accuracy. This allows for the correction of genetic defects, the introduction of beneficial mutations, and the development of new gene therapies. For example, researchers are using CRISPR-Cas9 to develop treatments for genetic diseases such as cystic fibrosis, sickle cell anemia, and Huntington's disease. By correcting the faulty genes that cause these diseases, CRISPR-Cas9 offers hope for cures that were once considered impossible. Beyond correcting genetic defects, CRISPR-Cas9 can also be used to introduce beneficial mutations into organisms. This has the potential to create new varieties of crops with improved yields, enhanced nutritional content, and increased resistance to pests and diseases. Section 6. Beyond the lab real-world applications of CRISPR. CRISPR technology is rapidly moving from the lab to the real world, with applications in healthcare, agriculture, and beyond. In medicine, CRISPR-Cas9 is being used to develop new treatments for cancer, genetic diseases, and infectious diseases. Clinical trials are underway to test the safety and efficacy of CRISPR-based therapies for a variety of conditions. In agriculture, CRISPR-Cas9 is being used to engineer crops with improved traits such as increased yield, enhanced nutritional content, and resistance to pests and diseases. CRISPR-edited crops have the potential to enhance food security, reduce reliance on pesticides, and improve the sustainability of agriculture. Beyond healthcare and agriculture, CRISPR-Cas9 has applications in biomanufacturing, environmental remediation, and even the development of new biofuels. The versatility of CRISPR technology makes it a powerful tool for addressing a wide range of global challenges. Section 7. CRISPR in Healthcare, a future of medical marvels. CRISPR-Cas9 is poised to revolutionize healthcare, offering new hope for treating and even curing a wide range of diseases. Researchers are exploring the use of CRISPR-Cas9 to correct genetic defects that cause inherited diseases, engineer immune cells to target cancer cells, and develop new antiviral therapies. One promising area of research is the use of CRISPR-Cas9 to treat genetic diseases. By correcting the faulty genes that cause these diseases, CRISPR-Cas9 offers the potential for cures that were once considered impossible. Clinical trials are underway to test the safety and efficacy of CRISPR-based therapies for diseases such as cystic fibrosis, sickle cell anemia, and Huntington's disease. CRISPR-Cas9 is also being investigated as a potential treatment for cancer. Researchers are exploring the use of CRISPR-Cas9 to engineer immune cells to recognize and destroy cancer cells. This approach, known as CAR T-cell therapy, has shown promise in treating certain types of blood cancers. CRISPR-Cas9 has the potential to transform agriculture, leading to more sustainable and efficient food production. Researchers are using CRISPR-Cas9 to engineer crops with improved traits, such as increased yield, enhanced nutritional content, and resistance to pests and diseases. CRISPR-edited crops could help address global food security challenges by increasing crop yields and reducing food waste. By engineering crops that are more resistant to pests and diseases, CRISPR-Cas9 could reduce the need for pesticides, benefiting both human health and the environment. CRISPR-Cas9 can also be used to enhance the nutritional content of crops, creating varieties that are richer in vitamins, minerals, and other essential nutrients. This could help address malnutrition and improve public health. While CRISPR technology holds immense promise, it also raises ethical concerns that must be carefully considered. One of the most pressing concerns is the potential for unintended consequences. CRISPR-Cas9 is a powerful tool, and off-target edits, where the system cuts DNA at unintended locations, could have unforeseen consequences for human health or the environment. Another ethical concern is the potential for genetic enhancement. CRISPR-Cas9 could be used to enhance human traits, such as intelligence, athletic ability, or physical appearance. This raises questions about equity, access, and the very definition of what it means to be human. The ethical implications of CRISPR technology are complex and multifaceted. It is essential to engage in open and informed discussions about the potential benefits and risks of this powerful technology. The future of CRISPR technology is filled with possibilities. As research progresses, we can expect to see even more innovative applications of CRISPR-Cas9 in healthcare, agriculture, and beyond. CRISPR-based therapies for a wider range of diseases are likely to emerge, offering new hope for patients and their families. In agriculture, CRISPR-edited crops with improved traits will become more commonplace, contributing to a more sustainable and resilient food system. CRISPR-Cas9 could also be used to develop new biofuels, create new materials, and even engineer organisms for environmental remediation. The CRISPR revolution is just beginning. As we continue to explore the potential of this transformative technology, we must proceed with caution, ensuring that its development and application are guided by ethical considerations and a commitment to the well-being of all. CRISPR technology has ushered in a new era in genetics, offering unprecedented power to manipulate the very building blocks of life. From curing genetic diseases to engineering drought-resistant crops, CRISPR-Cas9 has the potential to address some of the world's most pressing challenges. As we embrace the CRISPR revolution, we must proceed with both enthusiasm and caution. We must ensure that this powerful technology is used responsibly, ethically, and for the benefit of all. The future of CRISPR is in our hands, and it is up to us to shape its trajectory for the betterment of humanity and the planet.